Biotechnology encompasses both the traditional view and the modern molecular technology. So it's a combination of what? It's a combination of traditional as well as modern molecular biotechnology. There are two types of bioreactors. One is called simple stirred tank bioreactor. The other one is called sparged tank bioreactor. Two set of oligonucleotide chains are added to the medium. They form complementary base pairing at 3' end of DNA template. Hello everyone, welcome back to the revision class of 2nd PUC Biology chapter 11 called Biotechnology Principles and Processes. So today in this particular revision class, I am going to discuss about the weightage of marks for this particular chapter from examination point of view and the type of questions that may appear in the board examination. What is the weightage of marks for this particular chapter called Biotechnology Principles and Processes? The weightage of marks is 6 marks, whereas 1 marker question you will get 1 question and 2 marker question 1 question and 3 marker question 1 question. So what is the distribution of the 6 marks? That is 1 marker question 1 question, 2 marker question 1 question and 3 marker question 1 question. So this is the weightage of marks for this particular chapter 11 called Biotechnology Principles and Processes. Moving on to the chapter introduction. What is biotechnology? The very word itself will tell you. Application of modern techniques to the field of biology is called biotechnology. So what is the definition of biotechnology then? Biotechnology is the scientific manipulation of genes, of manipulation of living organisms, especially at the molecular genetic level, to produce what? To produce new products like what? Like hormones, like vaccines and monoclonal antibodies, which are beneficial or which are useful to mankind. Now, how do we define the word called biotechnology according to EFB, that is European Federation of Biotechnology? What is the definition? It is the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. So what is the definition according to EFB? EFB stands for what? European Federation of Biotechnology. It is the integration of natural science, that is natural science and organisms, their cells, parts thereof, and molecular analogues for products and services. In other words, you can call it as an industrial exploitation of any biological system, any living cell, in order to obtain what? Useful. We take the living organisms and extract some useful products for the welfare of human beings. The term biotechnology, who coined the word called biotechnology? The term biotechnology was coined by Carl Erecki. It is called Carl Erecki. Carl Erecki, a Hungarian engineer in 1919. The term biotechnology was coined by Carl Erecki, a Hungarian engineer in 1919. So when we say biotechnology, how is it? It is derived from the fusion of biology and technology. As I said, it's an integration of biology and the modern techniques what we apply to the living organism to extract products which are useful to mankind. Biotechnology encompasses both the traditional view and the modern molecular technology. So it's a combination of what? It's a combination of traditional as well as modern molecular biotechnology. What are all the important things you are going to study in this particular chapter? What are the important concepts? That is the principles of biotechnology. Very important is genetic engineering, manipulation of genes. We can alter the gene structure to get a desired product. Then maintenance of sterile ambience, sterilization is very, very important. Then what are the tools of recombination DNA technology? We use the word called rDNA technology. We have restriction enzymes, that is REN, restriction enzymes, cloning vectors, and competent hosts. Then what is the process of recombinant DNA technology? What are the steps that are involved in rDNA technology? That is isolation of the genetic material, cutting of DNA at specific location, by RNA. Hence, we call RNA as molecular scissors, which cut the DNA at specific sites. Then insertion of recombination DNA into the host cell, then obtaining the foreign gene product, then downstream processing. All these are the very important steps involved in the process of rDNA technology. Next, let us move on to the study of what will be the type of questions that are going to be asked from the examination point of view. That is, let us discuss the one marker question first. Define recombinant DNA technology, or short form is what? rDNA technology. What is it? It is in vitro process where hybrid DNA is created by combining the DNA from two or more sources to achieve desired genotype and phenotype in an organism. So the very word itself will tell you recombination means what? In vitro process where hybrid DNA is created by combining the DNA from two or more sources to achieve desired genotype and phenotype in an organism. 2. Restriction endonuclease is also known as molecular scissors. So REN, as I said, there are two types of REN, endonucleases and exonucleases. So restriction endonuclease is called as molecular scissor, which cut the DNA at specific sites. So why it is called? RNA cut the DNA molecule at specific restriction site. Hence it is called as what? REN, restriction. It is called as endonuclease, or it is called as molecular scissor. Then DNA, if RNA is called molecular scissor, ligase is called as molecular stitcher, or it acts as a glue. So DNA ligase is also known as molecular glue. Why? Because it is used to join or stitch together two DNA strands of desirable gene and cloning vector. So DNA ligase is also known as what? Molecular glue. Why? Because it is used to join or stitch together two DNA strands of desirable gene and cloning vector. So what is DNA ligase? Is also known as what? Molecular glue. Why? Because it is used to join or stitch together two DNA strands of desirable gene and cloning vector. Now, what is polymerase chain reaction, or it is called PCR? What is it? It is in vitro technique used to make copies of DNA segment using Taq polymer. Taq stands for what? Thermus aquaticus, which is a bacterium. Thermostable, and Taq polymerase is a thermostable enzyme. So what is polymerase chain reaction? It is an in vitro technique used when the DNA sample is very less. So in that, used to make particularly when this technology is used in forensic science. So used to make copies of DNA segment using Taq polymerase, that is Thermus aquaticus, a bacterium, a thermostable enzyme. Or when available DNA sample is less. At the site of crime, maybe the DNA sample is very less. So it can be multiplied. So by this technology, then it is amplified by the technique known as DNA amplification. Next, what is cloning vector? These are the DNA molecules that can carry foreign gene and replicate inside the host. Hence the name, they are called as what? Cloning vectors. Define plasmid. The very word, so the plasmids are used as what? Vectors. So they are nothing but extracircular, double-stranded circular DNA present in the cytoplasm of a bacterial cell which undergo autonomous replication. Next, name the plasmids used in rDNA technology. What are the types of plasmids that are used in rDNA technology? One is called PUC-18. Expand PUC-18, plasmid of University of California. PBR 322, P stands for plasmid of, B stands for Bolivar, and R stands for Rodriguez. This is the strain, type of strain. Then TI plasmid. TI stands for what? Tumor inducing plasmid. So name the plasmid used in rDNA technology. PUC-18, that is plasmid of University of California. PBR 322, that is plasmid of Bolivar and Rodriguez. TI plasmid, that is tumor inducing plasmid. Name the commonly used vectors for transformation in plant cells. So which is the commonly used vector? Even Agrobacterium tumefaciens, which induces what? Crown gall bacterium. Next, define gene gun. Gene gun is also called as microprojectile gun or shot gun or biolistic technique. So what are the alternate names for gene gun? Microprojectile gun, or it is called shot gun or biolistic gun. What is it? Insertion of DNA by coating with 1 to 2 millimicron gold or tungsten particles and bombarded with high velocity, hence the name gun, bombarded with high velocity helium pressure particle gun to the target cell, that is the plant cell. Now, what is a bioreactor? So there are two types where the biological reaction takes place in this large container called bioreactor. There are two types of bioreactors. One is called simple stirred tank bioreactor. The other one is called sparged tank bioreactor. So what is a bioreactor? It's a large vessel for growing organisms such as bacteria, yeast having rDNA and used to produce pharmaceutical compound or waste recycling are known as what? Bioreactors. Next, for 2-marker question. Mention the tools required for rDNA technology. This is for the type of questions which might be probably asked for 2-marker questions from this particular chapter called biotechnology, from this particular chapter, right? So, mention the tools required for rDNA technology. Desirable gene or genes, or we can also use it, call it as alien DNA or transgene or foreign gene or introduced gene. Then enzymes, cloning vectors, host cells and bioreactors. Next, what is palindrome sequence? Give an example. For example, we can give Madam, Gadag, Malayalam, so what is it? When you read from both the ends, it means the same. Madam, M-A-D-A-M. Here, M-A-D-A-M. Gadag, G-A-D-A-G, G-A-D-A-G. Malayalam, M-A-L-A-Y-A-L-A-M, M-A-L-A-Y-A-L-A-M. So what is it? What is palindrome sequence? Give an example. It is 4 to 8 nitrogenous base sequence on DNA molecule which is pronounced same. I told you it is pronounced, I just gave an example. Pronounced same when read in forward and backward directions on both DNA strands. It is also known as recognition site or restriction site. For example, you can take phi dash GAA-TTC 3 dash. From when you read forward like this, read from backward it will be what? Phi dash GAA-TTC. So forward and backward it is pronounced the same. Such type of sequence is called as what? Palindrome sequence. Next question, third question. Describe briefly downstream processing. This is a very common question which is asked in the board examination. What is downstream processing? Separation and purification of biosynthetic product obtained from rDNA technology is called downstream processing. So what is it? Separation and purification of biosynthetic product obtained from rDNA technology is called downstream processing. During the process, preservatives are added. In case of drugs, clinical trials are conducted. As you know, whenever a drug is found, clinical trials are done, maybe on animals. Then quality control testing is conducted. Next, name the host cell used in genetic engineering. Like we have E. coli, that is Escherichia coli, yeast cell, Bacillus subtilis and Xenopus oocyte. Name the non-vector mediated method of gene transfer into host cell. So what are the non-vector, without using any vectors, mediated method of gene transfer? It is, one is called microinjection, second one is called electroporation and the third one is called gene gun technique. 3-marker questions. So what will be the type of questions asked for 3 markers? Mention the different steps and of process of recombinant DNA technologies. Extraction of DNA, fragmentation of DNA using REN, isolation of desirable gene, amplification of gene using PCR technique. As I said, if the DNA sample is very less, you can amplify it through PCR technique. Ligation of DNA fragment into vector using DNA ligase, transferring rDNA into host cell, culturing the host cell on suitable medium in large scale, then extraction of desirable products. So all these are the very important steps during the process of rDNA technology. Then explain the steps of PCR technique, that is polymerase chain reaction. First one is denaturation, second one is annealing, third one is extension. That is, what is denaturation? Double stranded DNA is made single stranded by heating DNA are known as what? Separated as what? Template strand, original strands. Then annealing, two sets of oligonucleotide chains are added to the medium. They form complementary base pairing at 3' end of DNA template. Annealing is done at temperature around, what is the temperature that is required here? That is 50 degrees centigrade. Extension, deoxyribonucleoside triphosphate, that is DNTP, are added to the medium. Taq polymerase, that is Thermus aquaticus, that is an enzyme, catalyze the polymerization reaction of nucleosides extending from primers towards 5' end of template. Taq polymerase catalyze the polymerization at 74 degrees centigrade, and number of copies of DNA obtained after are n number of cycles. So you will get n number of DNA fragments through this technique called PCR technique. Moving on to the next question, that is the 3-marker question. Describe briefly the bioreactor. Discussing about the 3-marker question, describe briefly the bioreactor. Here you can see the diagram of a bioreactor. This is simple stirred tank bioreactor. As I said, it's a vessel where biological reaction takes place. So there are two types of bioreactors. One is called the simple stirred tank bioreactor, which you are seeing in the diagram. The other one is sparge tank bioreactor. So here it is divided into body, inlet, agitator system, then oxygen delivery system, a foam control system, pH control system, and temperature control system, pressure control system, sampling ports. Now let us study one by one. That is body. What is this body? That is the bioreactor made up of. Bioreactor is made from corrosion resistant material which can withstand repeated sterilization with high pressure steam. So the body here. Next, inlet. Inlet to introduce the microbes and nutrient medium into the bioreactor along with the passage of steam. Here you can see, along with the passage of steam. Agitator system, here you can see the blades, flat bladed impeller. These are sterile stirrer that facilitates the mixing and oxygen availability throughout the bioreactor. Next, oxygen delivery system. That is part. It is a tube with small holes. It releases sterile air bubbles into the bioreactor. Here you can see the sterile. This is the oxygen delivery system. So what is it? It is a tube with small holes. It releases sterile air bubbles into bioreactor. Then a foam control system. Here you can see the foam control system. It is used to introduce anti-foaming agents to prevent foam formation during the production of proteins in bioreactor. Then pH control system. Here you can see the pH control system. It is an optimal inlet to introduce alkali into the bioreactor to maintain pH inside the bioreactor. Then temperature control system, cooling jacket, you can call it as the cooling jacket. Bioreactor is provided with water jacket which maintains constant temperature in the bioreactor. Pressure control system, bioreactor is provided with water pressure gauge which is used to maintain constant pressure in the bioreactor. Next comes the sampling ports. It is used for periodic. So periodically the products are removed from the biotechnology, products from bioreactor. That is, they are removed with aseptic conditions to get the desired biotechnological product. So you have to describe with a neat label diagram for the bio, that is if at all a bioreactor is asked. At the end of this chapter called Biotechnology, Principles and Processes, what are the things you are going to learn? Define and explain the principles of biotechnology. Mention the basic techniques in creation of rDNA. First, the definition of biotechnology according to EFB. Then enumerate the tools of recombinant DNA technology. Describe in detail the naming and functioning of restriction endonucleases. Describe the principle and use of gel electrophoresis. Explain cloning vectors and the features that are required to facilitate cloning into a vector. Explain the need for a competent host and the methods of introduction of rDNA or alien DNA into the host. Describe the structure and functioning of the commonly used bioreactors and their use in producing desired gene products. Explain the steps and the need for downstreaming processing. So all these are the learning outcomes of this particular chapter called Biotechnology Principles and Processes. Let us have a brief recap with the flow charts. So different steps in rDNA technology, stages in rDNA technology: isolation of DNA, cutting of DNA at specific locations, isolation of desired DNA fragment, amplification of the gene of interest, ligation of DNA fragment into a vector, insertion of rDNA into host cell organisms, recurring the foreign gene products. Then types of bioreactors, I has already said. What are the types? One is called simple stirred tank bioreactor. The other one is called sparged stirred tank bioreactor. Then principles of the biotechnology, very important. It is based on what? Two very important aspects: genetic engineering and sterilization, maintaining of a sterile ambience. Then tools of rDNA technology, enzymes like restriction enzyme, DNA ligase and lyases. Cloning vectors like plasmids and bacteriophages. Competent host organism like through heat shock, microinjection and gene gun. So tools for rDNA technology, you can classify them into enzymes, cloning vectors and competent host organisms. So enzymes, restriction enzymes, DNA ligases and lyases. Cloning vectors, plasmids and bacteriophages. Competent host organisms through heat shock, microinjection and gene gun. Hope you have understood the important concepts, a brief discussion of the concepts of this particular chapter from the examination point of view of second PUC biology syllabus, chapter 11 called Biotechnology Principles and Processes. Thank you.